Welcome to Taconic's webinar, which today is entitled Rat Cancer Models in Oncology and will be presented to you by Dr. Marc Hillery de Boisferrand of Oncodesign, which is Taconic's collaboration partner in the field of oncology. First, please allow me to introduce myself. My name is Holger Kissel, and I will be your moderator today. Before we get started, I'd like to address a few practical matters. This webinar will be made available to all attendees. We will email a link to the presentation to you by the end of this week. Directly after the presentation, Marc will answer your questions as they appear in the control panel. So, if you have a question, please type it into control panel. The other attendees will not be able to see your question as it is sent directly to Marc and myself. If you cannot see the control panel, just single-click on the single arrow on the top of your screen. In case your question is not answered at the end of today's session, Marc will respond to you within the next 24 hours via email. If you are experiencing technical difficulties, please send the host a chat message and our staff will be able to assist you. So we want to get started now, and we hope you will enjoy today's webinar about the use of rat cancer models in the field of oncology. First, I want to introduce Marc Hillery de Boisferrand. Marc obtained his PhD in pharmacology from the University René Descartes in Paris, in France. Marc has now been working for the past 10 years as a study director in preclinical experimentation at Oncodesign. He is now the head of the in vivo pharmacology lab and continues to supervise a large panel of studies for Oncodesign clients. Since his arrival at Oncodesign, Marc has significantly contributed to developing new tumor models and metastasis models in mice and rats, and has played a central role in the development of the patient-derived xenograft platform that is available at Oncodesign. So, without further delay, I'm handing this presentation over to Marc, and Marc, we're all listening now. Thank you very much.
Thank you. Hello, everybody. In this presentation, I'm going to speak about rats in oncology. Rats and mice are both rodent species. However, despite the fact that these two species have a similar appearance, there are significant differences between rats and mice as disease model in oncology. In the first part of this presentation, I am going to describe available rat tumor models for anti-tumor activity. And in a second time, I'm going to describe the advantages of rats as model for oncology. And there are many areas of investigation for which rats are easier to use in comparison to mice. Rats have been the major model species used in several biomedical fields, and particularly in toxicology. There has been a long history of using rats to study cancer. The use of rats as cancer biology models began over a century ago. The first transportable tumor system was developed in 1910. Since major efforts were performed by scientists in the science of preclinical models in order to test a larger number of drugs. Significant efforts were particularly performed in the investigation of more refined tumor models. There is 5 families of available tumor models: the Syngeneic models, the Xenogenic models, the Orthotopic models, which can be Syngeneic or Xenogenic, chemically induced models, or transgenic models. The refinement of these families of tumor models are in opposition to the use in preclinical anti-cancer drug evaluation. The advantage of refined tumor models is firstly the fact that these models are more clinically relevant. The disadvantage is that these tumor models are more costly. A second issue is the availability of these refined tumor models and the necessity of high technique competency. The Syngeneic tumor models are a rat tumor which grow in a rat strain in which the tumor is originated. The graft is performed subcutaneously, and the graft is issued from available rat tumor cells or rat tumor fragment. The stage of this tumor development of this tumor is late and correspond to invasive cancer. In these tumor models, the research on early stage of tumor development could not be performed. The advantages of these tumor models is that they are relatively low cost and reproducible. For this family of tumor models, the host is immunocompetent, and working in a complete biosystem is a big advantage to study interactions of drug with the immune system. A second advantage is to use tumor cells and consequently use cells with a strong drug response database or with a capacity of rapid getting of robust in vitro results. Moreover, the hosts are easily available. The disadvantage is that the pathology is an animal pathology. Host and graft have same genetic background, and that cell lines express rat antigens or rat targets. More and more therapies are now targeted therapies like antibody or specific kinase. In such tumor models, it is necessary to work with animal-specific target and not human target. And the work on human-specific target is a major issue of this kind of tumor models. And this issue can become a serious limiting factor for Syngeneic tumor models. At Oncodesign, we propose a large variety of Syngeneic tumor models on Wistar, BD9, Fisher, and Copenhagen rats issued from bladder, brain, colon, ovary, or prostate tumor cell lines. The table shows non-exhaustive example of classic grafted tumor models. A full list is available upon request. In opposition of Syngeneic tumor models, we can propose Xenogenic tumor models. The development of human tumor xenograft was a major discovery in the field of using tumor models more clinically relevant. As already described for Syngeneic tumor models, the stage of tumor development is late. One disadvantage is to use immunodeficient animal able to be xenografted. At the difference with mice for which a lot of immunodeficient strains are available, like skin mice, not skid or nog mice, for rats, only nude rats are available. The nude rats are similar to nude mice with a genetic mutation that causes a deteriorated thymus, and this mutation resulting in an inhibited immune system and no T-cell development. For this family of models, the pathology is human. The stromal component of the tumor is rodent. As described, the host is immunoincompetent, and consequently, research are not performed in a complete biosystem. Available animals are more costly, and specific husbandry for animal stimulation is needed. Principal advantage is to use malignant cells, and there is a large variety of available cells. Again, these cells, we have a strong drug response database. A big step in the development of xenografted tumor models was due to the arrival of the new mouse strain in 1966. This strain is atemic without T cells. After that, new immunodeficient mice were described, the skid mouse, without T cells or B cells, the skid beige without T cells or B cells and an impaired NK cell function, the node mouse without T or B cells with an impaired NK cell function, an impaired macrophage activation, and a lack of hemolytic complement. And the NOG mouse strain, the NOG mouse strain without T, B, or NK cells with an impaired macrophage activation, a lack of hemolytic complement. This strain is the most immunodeficient mice. Compared to new mice, using more immunodepressive mice, an engraftment of PBMC, peripheral blood mononuclear cells, or human stem cell lead to new rodent models with the ability to be xenografted with tumor in a reconstituted immune system. The nude rats appeared in 1980, and no more immunosuppressive rats are available. At Oncodesign, we can propose a large variety of xenografted cells on nude rats. It is known that nude rats are less permissive than nude mice. However, because of our robust technology, 90% of models developed on mice are transplantable on nude rats. In the scale of the description of refined tumor models, there is orthotopic tumor models. Contrary to subcutaneous tumor models, in which the tumor grows in a non-natural seat, a more refined model is to implant tumor cells or tumor fragment in the seat of the primary tumor from which the tumor cell line is derived. A large variety of organs could be grafted: bladder, breast, by the mammary fat pad injection, colon, kidney, etc. For these models, the stroma is still rodent. The host could be an immunodeficient or immunocompetent animal with advantages and disadvantages as already described. The major advantage is that this family of model is more appropriate to mimic the clinical pathology. The disadvantages are the complexity of surgery. It should be noted that this issue is major for mice compared to rats, and this because of their large body size. However, even in rats, the surgery is complex and then lead to the use of a limited number of rats per experiment. For example, a stereotaxic injection of tumor cells needs around 50 minutes per rat. Another difficulty is the endpoint of orthotopic tumor models. The tumor growth is internal, excepted for mammary fat pad injection, and then difficult to follow. And the endpoint is often only the survival of animals. To overcome this difficulty of the endpoint, costly material is necessary, like MRI or PET apparatus, and apparatus dedicated to rodent. For example, it is possible to follow the growth of glioma models in the brain. We see here the tumor, and this by MRI, and this before the observation of parallel or indirect macroscopic signs of tumor development like the body weight loss. Moreover, it is possible to observe the brain tumor volume and then appreciate the anti-tumor activity of compounds before waiting the animal death, and then wait until the end of the complete rat mortality. In this example, before the first death of animal in vehicle-treated group, the anti-tumor of temozolomide in comparison with vehicle or radiotherapy is without any ambiguity. The MRI results observed here are in correlation with the survival. At Oncodesign, a platform dedicated to animal imagery is available. Other quantitative endpoints could be determined, like dosage of circulating tumor biomarkers or like carcinoembryonic antigen or PSA. For chemically induced rat tumor models, the major advantage is to work at all stages of tumor development, since the genetically altered cells until the late stage of tumor development. Moreover, the experiments are performed in a complete biosystem with a complete immune system. The tumor stroma and the pathology are rodent. The animals could be followed during a long period of time, and the most appropriate treatment period can be determined. Moreover, drugs could be studied through preventive or curative treatment approach. The disadvantage is to generate a sufficient number of animals at the same stage of tumor development and the heterogeneity of these models. However, this heterogeneity is clinic reality. At Oncodesign, we can propose different number of chemically induced models like colon, breast, or liver. As described for colon and liver, the tumor growth is difficult to follow, and MRI imaging is an interesting added value. The GmbH-induced breast tumor development could be measured with a caliper. The major interest of these tumor models is the hormonal dependency of tumors. This tumor could be studied without estradiol supplementation. Here we can see control group with the tumor growth, and here the ovariectomy treated group. Similar models are the transgenic models, in which the pathology is human-specific. The key benefit compared to chemically induced tumor models is that these models mimic exactly the human pathology and exactly the same mutations found in humans. These models will certainly play a large role in oncology. However, the number of available transgenic tumor models is still a limit. One example is a pig rat that carries a knockout allele of the APC gene. The mouse knockout models, the APC mean, which have been largely studied, develops primary tumor in the small intestine. The knockout rats develop colonic tumor similar to human patients. However, the difficulty is to generate sufficient number of animals at the same stage of tumor development. Of course, these models are highly raffined and make transgenic rats attractable tools for specific experimental manipulation and translational research. Similar technique could be used in clinic and preclinical studies. I am now going to speak about the benefits to use rats as cancer models. First of all, I will describe morphological characteristics of rat cancer model. Regarding an example of primary tumor architecture, the stroma component is 1.5 to 2-fold higher in rat compared to mouse. Regarding anatomopathology analysis, the stroma component in rat tumor model better mimic the human pathology. In the same manner, it is known that the tumor plasma volume in rats is higher compared to mice. The tumor in rats had a significantly greater catrons compared to mice. The catrons represent the permeability of the tumor. Consequently, with these higher catrons in rat tumor models, a better drug or tracer uptake could be observed. This point will be described in next slide. In the same manner, because of this higher catrons parameter, the tumor models on rats are appropriate tools for studying anti-angiogenic and vascular targeted drugs. With an higher catrons value, a decrease of this parameter could be better appreciated. Relative to the fact that rat tumor models are appropriate to study anti-angiogenic compound, at Oncodesign we have performed a study and test an anti-angiogenic drug. Here, we present the tumor volume measured with a caliper during the course of the experiment. At day 2, 48 hours after the initiation of treatment, we were able to observe a significant catrons decrease, whereas at the same day with a caliper, the anti-tumor effect of the compound was not still visible. Similar results were obtained at day 13. However, at this period of time, it was possible to observe a significant tumor volume decrease measured with a caliper. Using mice, this decrease of catrons value can be really appreciated. Moreover, as described, catrons could be determined in orthotopic tumor models, and in this case, without any AD of tumor volume evaluation. Moreover, it should be noted that the catrons parameters is used in clinic as an activity biomarkers, and results obtained in rats could be transposable to humans. As described for morphological characteristics of rat cancer model, for pharmacokinetics and pharmacodynamics analysis, there is a real advantage to use rats. First of all, the repetitive biological sampling can be performed. For example, you can see comparative pharmacokinetic profiles at different days of one test substance in tumor-bearing rats. Same rats were collected at each day of collection. The number of the parameter of the number of animals is considerable in the number of animal used for such experiment. For rats, there is a large choice of administration routes, and particularly IV infusion. Catheter is placed in the rat's femoral vein, and continuous infusion could be performed in parallel of blood sampling. The hepatic metabolism differs between mice and rats. It is known that the metabolism of rats is closer to human. For example, interesting pharmacokinetic profiles of everolimus suggest that the maximal concentration, the area under curve, and the half-life of compound in plasma attained by mice are considerably lower than obtained in humans. Whereas in rats, these parameters are closer to those attained in patients. It is well known that the more common application of rats in cancer research is to screen compounds for toxic side effects. Due to a similar metabolism and pharmacokinetic profile, a major advantage for anti-tumor activity pharmacological evaluation is the similarity between doses in rats and humans. As explained, because of this large choice of administration routes, it is possible to select the best route of administration and then compare toxicity profiles, anti-tumor activity profiles, and pharmacokinetic parameters by IV bolus route or IV infusion route, and that in the same biosystem. For example, compared to vehicle-treated rats by IV infusion, no toxicity was observed, whereas a difference in the body weight gain was observed between groups when the compound was injected by IV bolus routes, and this for a similar anti-tumor activity. Relative to the pharmacological evaluation, in this slide, we can see the pharmacological effect of vinorelbine in nude mice or nude rats. These two models bearing subcutaneous human prostate PC3 tumors. At their maximal tolerated dose, 10 milligrams per kilo for mice and 1.5 milligrams per kilo for rats, similar anti-tumor activity is observed. It should be noted that the dose injected in rats is close to the dose used in humans. It is known that there is no survival benefit of patients with metastatic prostate cancer after treatment with chemotherapy. In contrast of previous slide, in which an anti-tumor activity was observed in nude rats bearing subcutaneous tumors, in bone metastases induced nude rats, and this induction was performed after injection of human cell line by intracardiac route, vinorelbine induced only a moderate anti-tumor activity. And finally, all rats died with bone metastases. These results correlate with the clinical situation. Similar results were observed in nude rats bearing orthotopic human PC3 tumor prostate. Again, vinorelbine had no effect on the survival of tumor-bearing rats. However, it is interesting to see that at day 31, a transitory effect of vinorelbine on the tumor volume of the orthotopic tumor was observed. These MRI results are interesting and shows that it is possible to observe an anti-tumor activity without increase the survival of animals. Similar results were observed in orthotopic xenografted glioma models. Because of their larger body size, it is possible to observe late effect of compound. Here, the effect of herbitux. Interestingly, it was shown that on this primary colon tumor, the effect of herbitux was late. Here. Using mice, the effect of herbitux was not detected, and unfortunately, for ethical reasons, the mice were terminated because of their too high tumor volume. In contrast for rats, because of their large body size, a higher tumor burden is allowed. In this particular example, the effect of herbitux were observed on a tumor with a volume superior to 1,500 to 2,000 millimeter cube. As described, compared with mice and rats, there is a greater K-trans value. And higher K-trans lead to a better drug tumor uptake. This better uptake could be appreciated with PET imaging in subcutaneous human primary PAC 120 prostate tumor models xenografted in mice and rats. Comparing picture 1 and 3, a better tumor uptake is observed in rats. Moreover, this prostate tumor is known to be hormonodependent, and a tumor response is always observed after castration of male rats or male mice. Castration reduces the FDG uptake in tumor-bearing rats, picture 3 and 4. In contrast, the tumor uptake is not modified by the castration of mice. These results are certainly due to the low level of FDG uptake in the tumor of not castrated mice. Compared to mice, the FDG uptake is higher in rats, and then a decrease could be observed. To conclude, as described, rats have been the major model species used in several biomedical fields, particularly in toxicology. Over the past 2 decades, Oncodesign have accumulated a great experience in rat models dedicated to oncology. Oncodesign proposes a large number of models, subcutaneously or orthotopic models, Syngeneic, Xenogenic, or chemo-induced models. Moreover, since 2 years, Oncodesign proposes rat xenografted with patient-derived tumor. These models use tumor collected from patients at the time of surgery and directly implanted in rodent. The key benefit of use rats is that using these models, an easier translation to human is allowed, and this compared to any other rodent models. First of all, there is a big similarity between the dose used in humans and rats. Using rats, repetitive biological sampling is allowed. There is a large choice of administration routes, particularly for IV infusion. At Oncodesign, we are able to perform continuous infusion for more than 3 weeks. Compared to mice, rats are more robust with a longer lifespan. Their robustness is a serious advantage for major surgery. Another advantage is a larger body size for higher possible tumor burden, surgical access to organs for orthotopic graft, and visualization tools make rats a superior model compared to mice. Their metabolism and drug pharmacodynamics and pharmacokinetics profiles are closer to human. Using only nude rats is a limit in the use of more refined tumor models. Rats can be used in standard preclinical tumor models. Rats can be grafted with tumor cell lines for subcutaneously, orthotopically, or metastasis models. Rats can also be chemically induced. In the field of refined preclinical tumor models, rats can be grafted with primary tumors. The advance in genetic engineering and the ability to create rat knockout models with specific gene alteration, we can anticipate a serious increase of available, more refined rat models. Thank you for your attention.
Well, thank you very much, Marc, for this comprehensive overview of the technologies that have been established at Oncodesign using rat cancer models. I think that was very interesting, especially highlighting the differences between the rat cancer models and that you have just shown and the mouse cancer models that are used so widely in the industry. So we are proceeding now to the question and answer session, and I would invite everybody that is online right now to submit their question in the chat box if there are any questions. Throughout the seminar, my colleague has collected questions that came in, and Marc, there are 4 questions right now that we have collected, which I'd like to address to you. The first question that I'd like to ask and forward to you is, do rat cancer models and mouse cancer models have a similar behavior in terms of sensitivity, I guess drug sensitivity, and tumor growth?
there are differences between rat tumor and mice tumor, as the blood content, the stroma component, the tumor uptake, and the pharmacokinetics is different. So there is a difference in the sensitivity of tumors compared to one compound in mice or rats. The tumor growth is similar. However, for with using rats, the ethical endpoints is higher compared to mice. So you can perform a longer study using rats. And in terms of sensitivity, there is differences, and due to the pharmacokinetics parameter, etc., that I explained in the presentation.
Right. Yeah. Okay. Thank you. We have one more question here that's along the same line, the comparison between mice and rats, and the question is about the advantages of the APC transgenic rats that you have shown. Can you detail through examples the advantage of transgenic rats over mice?
I'm not a specialist of transgenic animals. At Oncodesign, we didn't perform a lot of studies on transgenic animals, and this certainly because of the animal cost. The chemo-induced rats are cheaper. It's tenfold cheaper for chemo-induced rats compared to transgenic rats, and there is the cost of animals and patent, etc. And there is not a lot of available rats models compared to mice, and this is certainly due to the genetic, the research in the genetic of the mouse. So I explained the APC transgenic, and there is another transgenic rat. It's the BRCA1 and 2 transgenic rats, and the major difference is that in mouse, this mutation, the gene mutation is lethal, and in rats, the rats are viable with this mutation, and so you can perform experiment on this mutation on the viable model.
Okay.
There is for the MSH6, the transgenic rats, the lifespan of transgenic rats are longer compared to mice. It's 1 year to 6 months for mouse, and after that, there is similarity in the development of cancer in animals.
Okay. Mark, we should proceed to there are questions coming in right now. I'd like to raise another question or ask you another question that came in, and that's a very practical question, I think. And the question is, do you have the genetic mutation status of your syngeneic models or expression profiling that I guess was performed on the tumors that are being grafted?
Can you repeat, please?
Yeah. Do you have the genetic mutation status of your syngeneic models or available expression profiling for these models?
No, there is no genetic mutation.
Expression profiling of the syngeneically grafted tumors, if that is available, or mutation status of the tumors. When
you engraft cells, there is a genetic mutation of this cell. So after, there is no differences in the cells engrafted and the cells in vitro. There is no more mutation in cell injected in syngeneic rats.
Okay, but just broadly speaking, I'm speaking for this listener now, do we have the mutation profile of the cancer cells that are being grafted in the rat?
No.
No. Okay. No. Okay. Good. I thought so. Yeah. We can be very concise then. Okay. One more question I'd like to ask from the audience is, I think this is something that you have addressed, but maybe we can just stress this again. What is the stroma content in the rat tumor?
It's the baseline of cells, and this stroma is rats. It's blood vessels and immune system of rats, and if you fibroblasts, etc.
Okay.
But
okay.
We didn't have performed the characterization of this kind of tumors.
Okay. Okay. Then another question that refers to the, I guess, the available rat lines that are available for grafting, and the question is, would there be an advantage to doing a xenograft on a rat that lacks both B and T cells? And would you expect, or could you graft a wider array of tumors in these rats? Maybe this would be also a good question to answer then, what rat lines are available in general in our field or what immunodeficient rat lines are available in our field for grafting tumors?
For when you want to work with a complete rat with a complete immune system, you have to perform Syngeneic grafts. So you work on graft pathology, and in the complete system you are similar to the clinic, but you work with animal pathology. And the difficulty is to work with immunodeficient animal, and with immunodeficient animals, you lose a part of the biosystem. So it's for that we are working with more immunosuppressive mice, and in the way to reconstitute the immune system in mice. And in the same animals, you can work with a reconstituted immune system, and you can engraft these animals. But for using new rats, for the times, you can't reconstitute the immune system.
Okay, thank you, Mark. Mark, maybe one more question then before we end this webinar. I just want to thank everybody who submitted questions. There are quite a few left to be answered, and that we don't get to your question today. We have collected all of them, and we will contact you via email, and Mark will provide the appropriate answers as much as we can do this. So the last question I want to raise to you that came in here is there or are there FDA regulations on what types of animal tumor models should be used in new drug discovery? I think it's a pretty big question, but maybe you can just based on your experience, do you know any specific, maybe for prostate cancer, for example, specific animal models that would be recommended by the FDA or would be regulated by the FDA for use in drug discovery?
For hormonodependent models, the best tumor models is on rats, because you don't need to supplement animals. For example, when you work with breast tumor, in mice, you need to supplement with estradiol, so you can't work on, for example, estradiol inhibitor. In rats, this is possible, and so for hormone-dependent tumor models, the best way is to work on rats. And after, for the clinical, for the evaluation of a drug, you are closer to those used in humans. So the rats are the best models for that, and there is not so much difference between rats to human compared to mice to rats. So if you want to perform more relevant preclinical studies and to expect a good translation to rats to human, it's better to work with rats.
Okay. Okay. Good. Thank you very much, Mark. I want to thank again the audience today for all of the questions that were submitted and also the comments that were submitted, especially around the PERG rats that was presented. Thank you very much.